Welcome to this genomic analysis workshop that's been brought to you by the Gregor Consortium in partnership with ClinGen. It's my privilege to get to tell you about the Gregor Consortium and how you potentially could collaborate with us. So the consortium stands for Genomics Research to Elucidate the Genetics of Rare Diseases. It was founded and funded by the National Human Genome Research Institute, NHGRI, to discover the causes of rare disease through innovative technology and vigorous collaboration. It was launched last fall, and the goal is to work with research participants that have suspected but molecularly undiagnosed Mendelian disease, that are consented for general research use or health or biomedical use of the data, that have high quality DNA that can be used to generate exome or genome sequencing data, that have been extensively phenotyped, and that data can be shared, and also the ability to recontact for deeper phenotyping. The data is all rapidly shared with the consortium and with other researchers through Anvil and dbGaP. So there are 6 centers total, 1 data coordination center at the University of Washington, along with 5 research centers at the University of Washington, Stanford, Baylor College of Medicine, Children's National in partnership with the diagnostic laboratory Invitae, and the Broad Institute. There are many goals of this consortium, and its overall goal is to improve genetic diagnosis and gene discovery, to define all the Mendelian disorders and catalog human clinical phenotypic impact of variants across genes. We want to drive new knowledge to the front lines by strengthening partnerships with diagnostic laboratories and developing tools that enable clinicians to increase molecular diagnostic and variant and gene discovery rates. There are many strategies undertaken. I'm going to highlight a few of them. One is working with unique populations, such as a cohort in Turkey that's led to the discovery of a lot of new genes and variants, understanding the role of structural variation in disease, functionally interrogating variants through a variety of methods, computational clustering of HPO data and other machine learning approaches, and then systematic reanalysis, because we're sequencing new data every day, while in parallel, we're also learning new information every week. And so we have large backlogs of data of cases that are still unsolved that we hope to be able to systematically reanalyze to improve diagnosis there. Different centers have different strategies of how they interact with collaborators. Baylor is trying to use a central consent across their center and adding different sites. The University of Washington Center for Rare Disease Research is another of the Gregor research centers. It's was also part of the Center for Mendelian Genomics, a previously funded program. And so they have over 10 years of gene discovery experience using exome sequencing. They collaborate with investigators and clinicians worldwide, focusing on new technologies and approaches to facilitate novel gene discovery. One approach is identifying moving into non-coding by looking for missing variants, particularly in cases where you have autosomal recessive disease, one pathogenic variant, and are looking for that second hit. That can be a really nice place to know where to look really deeply. They offer short-read exome and genome sequencing, long-read sequencing, RNA-seq, methylation profiling, comprehensive bioinformatic analysis, and experimental and functional validation. And I've included a short URL down there where you can actually propose a research cohort with them. That would be part of the Gregor Consortium. Overall, all the centers are using a number of approaches, including exome and genome sequencing, looking for a lot of different types of variation, different functional studies, long-read sequencing, and RNA-seq, and then sharing all of this through a number of strategies, including data sharing in Anvil, sharing variants in ClinVar, gene matching through Matchmaker Exchange, sharing rare variants in aggregate across the rare disease datasets and websites like Deno2MP, and sharing cell lines from functional modeling through various approaches. And so I've also shared information down here about how you can read about the requirements to work with another of the research centers through the Broad, and how you can propose a cohort there. There's many different multi-omic approaches that are being tried. This is some of the proposals from Stanford, where they're going to generate high throughput multi-omic data, including metabolomics, lipidomics, ATAC-seq, methylomics, RNA-seq, transcriptomics, all the omics. They're also doing long read. They're going to apply machine learning in these multi-omic approaches to try to best tease out which approaches are most effective in which settings, and also try to improve diagnostic rate and estimate diagnostic rate with different approaches. They'll be using natural language processing, reference for multi-ethnic genomic data, polygenic risk modeling, along with single cell reference data in developmental context. They also have an array of functional validation assays that they'll be using. And then all the centers, while we're all using many of these approaches, the other thing that's really important to all of us is increasing outreach and education. So we're trying to hold workshops like this. ASHG will often hold workshops. And then we're partnering with different researchers and patient advocacy groups to bring undiagnosed individuals into the research consortium, into the Gregor Consortium. We're building non-European collaborations in addition. And then we're training graduate students and postdocs through these centers in rare disease analysis, along with having many junior faculty being involved in these centers in the consortium. And so this is from Children's National Hospital in Vitae, which I think really summarizes the efforts of the Gregor Consortium overall, is that we want to discover new things, including new disease genes and variants that are not found on standard testing, particularly going beyond the exome. We want to disambiguate a lot of this data. There's a lot of variants of uncertain significance that challenge analysis. And so we'll have different approaches to do that, including functional modeling and transcriptomics. And then we want to disseminate this information through consortium data sharing and other efforts. So all of the centers will be working together, and we'll be doing this through vigorous collaboration and really adapting our approaches to figure out how to really work as a team and then partner with other groups around the world to really make a dent in improving the diagnosis rates. Now, we couldn't do any of this without the Gregor Data Coordination Center, which manages the data release of including developing the data models, harmonizing and QCing the data as we get ready to share it. They manage our program outreach, engaging with clinicians, families, and researchers. They coordinate the logistics of the Gregor Consortium, and they also develop and maintain the Gregor Consortium website, which is one of our current key ways we communicate with the community. And they'll be managing the Opportunity Fund, which are small awards to support the work of researchers from outside the consortium to follow up on promising findings or new approaches within the consortium. And so keep an eye out for a call for applications for the Opportunity Fund for Gregor, likely later this year. I can't share very many details about it because it's still being developed exactly what it will look like, but more information is on its way. So we are sharing all our data through Anvil. It's not yet there, but it's coming later this year. And I just wanted to highlight that there already are a lot of data sets already available in Anvil, including the rare disease data from the Center for Mendelian Genomics, with over 100 terabytes of data from exome and genome sequencing. And so you can apply for access to this through dbGaP. And then rather than having to bring the data locally, you can actually analyze it right there in Anvil with a lot of the tools that have been built in. And you'll hear a lot more about that later in this workshop. So there are many ways to currently get involved with the Gregor Consortium. I think these are going to evolve over time as this consortium further develops, but you can access data through Anvil. You can provide samples to the consortium through collaborative sequencing and analysis, and you can share candidates or apply for opportunity funds for follow-up of different candidates. So we're really excited about this workshop, which we're just kicking off, but there's another workshop I already wanted to mention to you, and that the UW-CRDR does have an annual summer workshop in August. That's 5 full days, a very much more limited attendance of 20 people, where they really focus on some of the computational approaches to rare disease analysis. And so I've included the website there to learn more about their workshop. So I wanted to thank you for your attention and just highlight some of the people that are involved in this consortium. I can only list the PIs here for the sake of space, but there are dozens, if not more, people involved in each of the centers. So this is a really big group, and we're really excited to be working together. The NHGRI program officers, Lisa Chadwick and Chris Wellington, are really leading the charge, and the individual PIs from each center are listed here. And you can read more about the consortium on our website, along with there's a form to get in touch with us if you have any follow-up questions. So it's a pleasure talking to you today, and I look forward to seeing you later in parts of this workshop. Thank you so much.